Good day, everyone. So we're reviewing Wetzel's Limnology textbook. This is edition number 4. We're on chapter 15, which deals with the phosphorus cycle. And today we'll be looking at benthic algae and phosphorus release from sediments. Oxygen is a primary factor influencing the release of phosphorus from sediments. If the sediments receive light, even at very low intensities, photosynthesis of epiphytic algal communities growing on the sediments can quickly produce high, often markedly supersaturated concentration of oxygen within the community, usually less than 2 millimeters in thickness. This oxygen can diffuse several millimeters into the interstitial water of the supporting sediments at rates greater than it is consumed by bacterial respiration and chemical oxidations, and thus suppress internal phosphorus loading. If respiration rates are high enough, superficial sediments can, however, become anoxic overnight. By this mechanism, diurnal changes occur in the oxidized microzone of sediments, shallow enough to receive light from fully oxidized in the daylight hours to fully reducing at nighttime. It was shown experimentally that the rate of phosphorus efflux from sediments is inversely related to the extent of sediment oxygenation and the magnitude of epiphytic algal photosynthesis. Although much of the reduced phosphorus efflux is caused by direct chemical redox changes, the microbial community also assimilates and complexes nutrients in organic compound. Phosphorus translocation by migrating phytoplankton. Several algae and cyanobacteria of lakes exhibit vertical migrations from nutrient-rich sediments or lower water strata to the euphotic trophogenic zone on a seasonal or daily basis. For example, a modal cryptonomad algae and a dinoflagellate may migrate vertically from phosphorus-rich strata below the metalimnion at night to surface waters in early morning in sufficient quantities to enrich the epilimnion. Migrations of a large part of certain benthic inhabiting cyanobacteria to the epilimnion have been implicated in the translocation of benthic phosphorus sufficient to increase phytoplanktonic productivity. Vacuolate cyanobacteria are able to regulate their position in the water column when turbulence is low, probably to optimize access to nutrients and light. They have been observed migrating upwards at 11 meters depth in a metalimnion and have been recorded migrating into anoxic waters, a distance that can be covered under low turbulence within several hours, depending on the species, cell size, and starting position in the water column. For example, maximum migration rates for Microcystis aeruginosa range from 0.4 to 10.8 meters per hour downward and 1.3 meters per hour upward, while Anabaena, recently renamed Dolichospernum, and Aphanizomenon downward and upwards were 0.04 and 0.14 meter per hour respectively. Phosphorus cycling mediated by aquatic plants and epiphytic microflora. Lakes, reservoirs, and rivers are coupled with their drainage basins by intervening wetlands and littoral zones, the land-water interface zones, through which much runoff and seepage water flows before entering the main water bodies. The aquatic plants of the interface zones are major biotic components in many, if not most, small lakes and in the floodplains of river ecosystems. These plants often occupy much of the epilimnion and trophogenic zone, both of which extend virtually to the same depth in many small lakes. The littoral macrophytes and their attached microflora function mutualistically as conservative retainers of phosphorus and can modify greatly phosphorus loadings and budgets among aquatic ecosystems. The importance of rooted plants to lake-wide phosphorus cycling will depend on the proportion of sediments receiving enough light to support plants. aquatic plants. Emergent, rooted, floating, leaved, and submerged higher aquatic plants take up most of their phosphorus from the interstitial waters of the sediments, where the concentrations are several orders of magnitude greater than in the overlying water. Even though the structural and vascular systems of submerged aquatic angiosperms are simplified and reduced, nutrient uptake is largely by the root rhizome system from the sediments. Early studies provided conflicting results. For example, the presence of the macrophytes aerocolon or utricularia in experimental sediment water systems increased the movement of radiophosphorus from the water to the sediments. Absorption and translocation of phosphorus studied in bacteria-free cultures of the macroalgae chara, common to hot water lakes, showed all parts of these algae could absorb phosphorus 32 about equally. About the same proportion of adsorbed phosphorus was translocated from the apices or from rhizoids to other parts of the algae. Similar results were obtained for other freshwater plants. Rates of phosphorus uptake and excretion by the roots and leaves of submerged macrophytes were found to be dependent on the phosphorus concentration of the medium. Because the interstitial sediment phosphorus concentration are very much greater than those of the water, uptake from the sediments predominates. Critical experimental studies of phosphorus uptake both from the sediments and from the water by submerged macrophytes that possessed their normal epiphytic microflora showed, however, that nearly all phosphorus was obtained from the sediments. Nearly all of the nutrients uptake from the water was by the attached microbiota, and very little was transferred to the supporting macrophytes. As foliage matures, a gradual partial senescence of leaves often occurs, particularly among submerged macrophytes from which nutrients leach. Some senescing leaves slough off the living plants and collect among detrital accumulations of the sediments. For example, the leaching of phosphorus from dead macrophytes under sterile conditions was rapid and resulted in a loss of from 20 to 50% of total phosphorus content in a few hours and 65 to 85% over long periods. Rates of leaching were greater from roots than from leaves. Macrophytes can also limit the release of phosphorus from sediments. Most aquatic angiosperms are herbaceous perennials with multiple cohorts. Upon completion of a growth cycle, a major portion, about between 25 and 75% of phosphorus of the foliage is translocated to the rooting tissues. Some of this phosphorus store can be permanently interred in senescing root tissues that decompose very slowly in anaerobic sediments. Oxygen release from roots of submerged macrophytes in anaerobic sediments can lead to iron cross formation and immobilization of phosphorus by iron-coated roots, impeding phosphorus release from sediments. Sediment resuspension is lower in macrophyte beds because of lower turbulence. Epiphytic microbiota. The surfaces of submerged vegetation are colonized with epiphytic microflora. The physiology and growth of attached microflora are coupled to the physical and physiological dynamics of the living substrata upon which they grow. The productivity of epiphytic bacteria and algae is very high. This community acquires nutrients both from the water within and passing through the littoral zone and from the supporting host macrophyte tissues. Although relatively little of the total phosphorus pool within actively growing macrophytes is released, this release can be important to the epiphytes. For example, certain algal species that grow adnate to the macrophytes can obtain over 60% of their phosphorus from the macrophyte. Even when the phosphorus concentration are very high in the water, some nutrients are obtained from the macrophytes simply because diffusion within the complex epiphytic community is too slow to meet demands. The phosphorus of the littoral water is very actively assimilated by the loosely attached epiphytic periphyton, incorporated into the periphyton and intensively recycled. The periphyton, rather than the submerged macrophytes, function as the primary scavengers for limiting nutrients such as phosphorus from the water. During dormancy phases and senescence of above-ground macrophyte tissues, releases of phosphorus are readily utilized by the periphytic community, which tends to develop profusively during autumn and winter periods. As the senescing macrophytes with their epiphyte communities collapse to the detrital mass near the sediment, much of the released phosphorus is rapidly retained and recycled by epiphytic bacteria and algae. Therefore, the attached macroflora, particularly epiphytes on submerged macrophytes, can function as an effective phosphorus scavengers from the water. The retention capacity is regulated by many environmental parameters and can be exceeded if the loading of phosphorus is very high or the rate of water flushing through this biological sieve is too rapid to allow time for the uptake and retention. Rapid water movements through littoral zones, such as by natural storm events or from artificial human-induced runoff, can result in flushing movements of nutrients into the receiving open waters at rates too fast to be retained biologically. Once phosphorus is cycling within the submerged macrophyte periphyton community, however, it is improbable that much of it will be exported to the open waters. So let's look at figure 15-9 here. Fluxes of phosphorus from the sediments to submerged littoral macrophytes and among the epiphytic macroflora of the periphyton. A. adnate algae, Al. loosely attached algae, B. bacteria, C. inorganic or organic particulate detritus, such as calcium carbonate. So this is the active phase, and this is the senescent phase. So this is where the macrophyte is dying and falling off, pretty much. So P is the phosphorus, and Al is the loosely attached algae, as well as adnate algae, and then B and C are bacteria, inorganic, and C inorganic or organic particulate. So we have this cycle going on here. We have done from the adnate to the loosely attached algae to the inorganic or organic particulate detritus, and then to the C, the inorganic organic particulate detritus. And then B, bacteria. So we have this cycle going on, and then the phosphorus coming in from the rooting zone of those plants. Now we have a senescence phase. So we have phosphorus coming in from dissolved fraction, although we have some that is lost as well. We have the same cycle going on in the leaf here, and then however phosphorus now is moving opposite direction than when it's an active system, so it's moving toward the roots, and then we have loss of some of the phosphorus from the rooting system to the surface of the sediment. And then we have, as the leaf senesces, we have loss of phosphorus through the senescence process. Littoral phosphorus fluxes and loadings. Major sites of phosphorus fluxes in lakes have been examined by dividing them into three general compartments. A, the pelagic open water and organisms of the epilimnion. B, the littoral macrophytes and attached macroorganisms. And C, the apolimnion and sediments. Early tracer experiments employing such compartment analysis indicated that phosphorus in the epilimnion is extremely mobile. The turnover time of phosphorus in the epilimnion of a small lake was found to be 3.6 days, within 20 minutes of the time it entered, over 95% of the added phosphorus was taken up by the plankton. It had a turnover time of less than 20 minutes. In Toussaint Lake, a small acidic lake with a well-developed littoral zone of rooted aquatic plants, the littoral region was the most important contributor to the turnover and retention of phosphorus in the epilimnion. Phosphorus was lost to this compartment 10 times more rapidly than to the hypolimnion and sediments, and 50 times more rapidly than its loss through the outlet. In comparable experiment in an acidic bog lake in Nova Scotia, with extensive development of littoral sphagnum moss, nearly all of the tracer phosphorus was taken up by the sphagnum and plankton. Essentially, no phosphorus reached the hypolimnion in the short term. Rates constants of phosphorus loss from the epilimnion cannot be derived from this data. However, reanalysis of the results of several studies indicated that the turnover time of phosphorus in the epilimnion ranged from 20 to 45 days and was inversely correlated with areas of the lakes and estimates of development of the littoral vegetation. In 200-liter aquaria with a steady-state flow of dechlorinated water and regulated steady-state inputs of phosphorus to the aquaria, attached filamentous algae in simulated littoral zones removed the majority of phosphorus before it reached pelagic algae in the epilimnion or open water. The equilibrium exchange between phosphorus input to or releases from the littoral flora and from ore to the epilimnetic water varies with the amount of phosphorus input from the drainage basin. Phosphorus uptake by and release from senescing littoral macrophytes and attached algae will vary with the physical constraints of littoral development determined in part by basin morphometry and water exchange patterns. This relationship can shift with seasonal changes in active growth of the littoral macrophytes. The physiology and growth of attached macroflora are coupled to the physical and physiological dynamics of the living substrata upon which they grow. By means of intensive recycling of limiting dissolved nutrients and gases to maintain high sustained growth and biomass, losses are minimized and important external nutrients can be directed primarily to net growth. Most aquatic plants are perennial with numerous cohorts and continuous growth, and turnover, particularly in regions of moderate climate where littoral growth is more or less continuous. Thus, the littoral complex of macrophytes and attached microbiota is often a major component of phosphorus retention and exchange with epilimnetic water, but is constantly changing in the effectiveness of nutrient retention and loading. Benthic invertebrates and the transport of phosphorus. Evidence for how effectively benthic invertebrates can influence the rates and direction of phosphorus exchange between sediments and the water has been controversial. Clearly, these organisms can enhance rates of transport of phosphorus from the interstitial waters of sediments under certain environmental conditions. In other cases, their influences are minor. Examination of the transport mechanisms related to benthic invertebrate activities or on phosphorus fluxes need to address feeding activities of the invertebrates, intestinal decomposition and excretion, feeding and respiratory behavioral activities that alter redox gradients in sediments and phosphorus solubilities, movements of organisms from the sediment habitat to other habitats with displaced phosphorus release, and their size and population density. This discussion only pertains to areas where there is sufficient oxygen to sustain benthic invertebrates. But benthic invertebrate behavior is not viewed as a major contributor of phosphorus to the water column because phosphorus transfer rates under aerobic conditions are relatively low and benthic invertebrates are absent under anoxic conditions. Benthic macroinvertebrates are extremely diverse in their feeding, respiratory growth, and reproductive behaviors. This diversity contributes to the conflicting, inconclusive, and occasionally uncritical conclusion drawn on the effects of organisms on phosphorus and nitrogen fluxes, simply because phosphorus may be transported from interstitial waters to water immediately above the sediments does not necessarily mean that these compounds will be transported in available chemical forms for use in the trophogenic zones of lakes or rivers. One must retain an ecosystem scale perspective in evaluating these fluxes. And I think that will be about it for today, and we'll pick it up tomorrow with this dominant macroinvertebrate groups right here. And thanks for attending today's session on phosphorus cycling, chapter 15 of Wetzel's Limnology textbook, and I'll see you tomorrow. Bye.